Gene writing is the ability to write small or short messages into a genome. And that might sound simple, it's really hard. And it's distinct from what we've called gene editing in several ways. Historically, gene editing has been based on an amazing category of machinery that in nature cuts or destroys DNA. And what we went looking for in creating Tessera is machinery that might have spent billions of years learning how to write DNA. And that category of machinery is called mobile genetic elements. They have an amazing scientific legacy. And it is close to being nature's universal vote on machinery with the ability to write. And we figured if we could get these to write therapeutic sequences and do so programmably, that would offer an extraordinary category of medicines. Tessera's been able to create 3 gene writing platforms, and those allow us to do things that have been either previously impossible or really difficult. So those include changing any one base pair to any other, A to a T, A to a G, A to a C, and every other direction. They include inserting and deleting sequences, and the ability to be able to write entire genes. And in that last category, we've been able to do something that's a first in the history of molecular biology, which is give just 2 RNAs to a human cell and have that cell wake up the next morning with a new DNA gene in its genome. And each of those categories of gene writing offer the opportunity for multiple applications in medicine. A natural home for many of our technologies is going to be rare genetic disease, but there are numerous opportunities in oncology and beyond. And given that DNA is the code of life that drives the biology of every cell in our bodies, every day of our lives, it is with certainty going to touch every therapeutic area. Some specific examples of where gene writers are going to have impact is we've got gene writers that can make the true correction of the sickle mutation that causes sickle cell around the world to the wild type allele, which all of us without sickle cell enjoy. That is one nucleotide, and unfortunately it's an alteration that previous gene editing technologies can't make. Our aspiration is to be able to make a sensational category of therapeutics for sickle cell. There are numerous other rare genetic diseases that we're active in as well. And then the ability to be able to write entire genes in the form of DNA using only RNA opens up some amazing doors for the category of genetic medicine, in that the same kind of compositions that us, our family members, our friends have now come to experience in the SARS-CoV-2 vaccine race, which are RNA payload, lipid nanoparticle delivery, have the potential to be a conduit through which to write corrective genes into the genome of cells throughout our body, and therefore open the door for cures for genetic diseases where the mutations driving that disease are very diverse and not just one, as well as to be able to impart means of protecting against disease ever occurring. In some ways, the closer you are to a first biology class, the more evident it is that genetic medicine is going to be a really big deal. And simplistically, DNA drives the biology of all of our cells all the time. And therefore, if you're trying to cure a disease or prevent disease from ever happening, DNA is going to be the rightful home for a big chunk of those therapies. So in some ways, you could think of it as being the final epoch of medicine, in that as we gain the ability to write in the code of cells around our body, the corresponding opportunity to prevent disease from ever occurring is vast.